Hello, everyone. Thank you and welcome to this 144th session of the International IVF Initiative and the 10th RBMO live session. My name is Mina Alikani, and I have the pleasure of hosting this session as an editor of RBMO. Before I introduce my esteemed co-host, I'd like to say a few words about our journal. Now, I might be slightly biased, but I think that RBMO is one of the most progressive and forward-thinking journals in our field. This character was instilled by Bob Edwards, who founded the journal 25 years ago, and it continues to be reflected in the type of papers that we accept to publish. We make extraordinary efforts to help our authors to improve and refine their work, and we showcase and recognize them in many different ways, like during these RBMO live sessions. Next year, we will be celebrating the 25th anniversary of the journal, and a special issue is being prepared for this occasion, which I'm sure our readers will enjoy. On behalf of the chief editors, Nick Macklin and Juan Garcia Velasco, I would like to invite you to submit your papers to RBMO and experience the journal firsthand. So now, today I am joined by my colleagues Basak Balaban and Thomas Ebner, both of whom are current section editors in the Clinical Embryology section of RBMO, and neither of whom really needs an introduction, though you can find their full bios on the I3 website. Basak is the head of the IVF lab at the American Hospital of Istanbul Assisted Reproduction Unit. She's a past president of Alpha Scientists in Reproductive Medicine, a past executive committee member of ESHRE. And of course, it is a great pleasure to have you here, Basak, and thank you for staying up late for this session. Thomas is an ESHRE certified senior clinical embryologist, a university professor in Linz, Salzburg, a former board member of Alpha and ESHRE, and he's also active in the ESHRE certification of clinical embryologists and the ESHRE Art Center certification for good clinical practice. It is wonderful to have you here, Thomas, and thank you for everything that you do for RBMO as a section editor.
Thank you very much, Mina, for this nice introduction. Dear colleagues, welcome to the RBMO Live and I3 session. It's my great pleasure to co-chair this webinar together with Thomas Ebner. And our host, Mina Alikani, I don't think she needs any introduction as she's very well known in our field, but I'll mention very briefly. Mina is a board certified high complexity lab director in assisted human reproduction with many years of experience in the field and consults on several IVF technological companies. She's an editor of Reproductive Biomedicine Online. And since 2012, she's serving as the president of the Alpha Society, the Scientists in Reproductive Medicine. It's a great pleasure to have you as our host, Mina.
Thank you so much.
And then I think it's up to me to also thank you, Mina, for having me on board. It's really a privilege and a pleasure to be part of this project. And I don't want to delay things, so let's go straight to the heart of the matter. And I'd like to introduce our first speaker, who is Michael Collins. And he graduated from Louisiana State University on continuous training by completing a postdoctoral fellowship at the University of Missouri in maternal nutrition and epigenetics. Most of his career has been in pharmaceuticals, in drug discovery and drug development. He currently oversees all sciences for DMRW Life Sciences in the USA. So please, Michael.
Thank you very much, Thomas. Appreciate that. On behalf of myself and my co-authors, I would like to thank RBMO and I3 for allowing us the time today to talk about the data that we've developed around the effort and manual operation of reproductive health cryostorage facilities. We had three objectives with this study. First was to apply traditional time and motion methodologies to human IVF cryostorage operations so that we could analyze the differences between study sites and how they correctly identify and safely deposit and retrieve specimens from storage so that we could then look for areas where automation and technology could eliminate or significantly reduce the need for physical and manual labor. So time and motion studies got started over 100 years ago with Frederick Taylor, who started proposing a time study as a way of operationally improving business processes. This was followed up by Frank and Lillian Gilbreth, who created motion studies in the 1920s and 1930s. They actually set themselves at odds with Frederick Taylor by promoting motion studies, or the movement required to complete a task, as the best way of identifying areas for process improvement. Over time, the two concepts did come together, especially in post-war rebuilding in Japan, where a time and motion study found its way into the Kaizen business philosophy. And today, time and motion studies are the foundation of many business improvement models, such as Six Sigma. For this study, we collected data from 4 sites, and the paper is termed Alpha, Beta, Gamma, and Delta. We had different code names for the sites during the data collections period. All of the data was kept confidential from all sites and from everybody else, with only a select few people knowing the identification of all of the sites and their specific data. If the sites requested, we did share their individual site data with them. We had 12 subjects total across the study, with 1 through 5 per site. Data was collected over a 2-week period, including weekends, at each of the sites. The study personnel were observers only. They did not interact with the work processes occurring. We also did not record any audio to make sure that there was no record of any patient information or anything else that could have jeopardized our IRB for this study. The data handling procedures were also recorded, and this included both physical and electronic processes that the data was handled with. There were also informal interviews conducted with the subjects and with laboratory directors to help us understand and be able to analyze the data in a better fashion. Following the data collection, site-specific process maps were developed for each site and each work processes. These were used to identify areas that were common to all sites so that we could then analyze those areas where automation and technology improvements could either eliminate steps or create more efficient cryostorage operations. The 4 data collection processes were specimen deposit, or the movement of a specimen from the benchtop to cryostorage facility, or specimen retrieval, the identification of a specimen within the cryostorage facility and movement of it back to the benchtop, what we termed a daily inspection and filling, but this is looking at liquid levels in the liquid nitrogen tanks, how the process is done to fill those liquid nitrogen tanks, and then finally the annual inventory. For the specimen deposits and retrievals, we targeted 12 live retrievals and deposits per site as our ideal number. Data was also not doubled if more than 1 embryologist was required to complete a task, as was common. Also, for the data inspection and filling, we looked at all of the processes involved, LN2 level determination, the fill process, plus, importantly, the data handling and the analysis of that data that was coming from the fill process. For the annual inventory, we asked the site to conduct an inventory over the period of 3 hours to see how much they could accomplish in those 3 hours, and then use that data to extrapolate how long it would take to inventory the entire site. For this study, descriptive statistics only were used. The disparity between the sites really limited the use of any inferential statistics. Looking at the results, we have here one of the process maps that was developed for a specimen retrieval from one of the sites. This is only the beginning of the process, where this site would stage their retrievals for the following day, the day before they intended upon using those retrievals. This is the second part of the retrieval of the specimens at this particular site. To give you an idea of how we did the analysis, the data on the next slide is only coming from these two steps right here, so that all of the other steps here were outside of this analysis that I'm presenting today. This analysis was looking specifically at the method of retrieval from pulling of a cork outside of the dewar to actually removing the correct cane. We saw across all of the 95 retrievals that we measured that it took either 2 seconds all the way to 10 minutes and 7 seconds to identify the correct cane and remove it from the dewar. We had 95 total retrievals, of which 89 were from small dewars. Of the 95 total retrievals, it took 3.4 queries of the data record for the location to correctly identify the cane that contained the specimens. Of the 89 retrievals from the small dewars, the canister was lifted into the neck 1.5 times, where, of course, only 1 time is required to retrieve the cane out of the canister. The time spent in the neck was 11.8 seconds on average, with the maximum time spent in the neck of the dewar being just less than 1 minute. We assigned a grade of 1, 2, or 3 to the lifting of the canister as to how high in the neck the canister was lifted, with the image on the left being a grade 1 and the image on the right being a grade 3. We see here that the average grade per pull was 1.4. We also looked at fatiguing work, and this was done over 8 hours of the embryologist working directly with cryostorage equipment. Of this 8 hours time, almost 1/2 of it, 47.25% of the time, is spent doing fatiguing work, which we defined as being bent over, lifting heavy objects, things that are greater than 2 liters in size, or using a stool to complete the task that they were performing. The majority of the fatiguing time is spent bent over, but there's still a hefty amount of time lifting heavy objects and also standing on a stool to complete a task. If you look at figure A there, you see somebody lifting a heavy object with their hand bent and are actually moving it to a cart that is visible in the bottom of the image. Time spent pushing or pulling carts was not included in this analysis. However, that was considerable that the embryologists were spending time working with various carts of different sizes and different weights. We also looked at the methods used to fill the dewars. Each site had a dramatically different method, with site alpha taking the tank to the dewar. Site beta actually ladled LN2 from a storage filled dewar into the dewars that had the specimens. Site gamma delta had buckets that they were using, and site gamma took the dewar to the tank. What we learned from this is that although it would appear based upon time that ladling is the most efficient, it is really dependent upon the physical plant requirements, with it being highly unlikely that ladling would be efficient for one of the larger IVF storage facilities that were included in our study. And likewise, moving a storage tank or a dewar to the tank would be overkill for one of the smaller centers that were in this study. So it's very dependent upon the physical plant requirements. In addition, we were unable to draw any main conclusions from this about what would be the most efficient way of filling the dewars when it comes to the manual operations. This study was limited, number one, by only looking at cryostorage related activities. There are likely many other efficiencies that are not captured in our study. Also, the heterogeneity of lab operations may limit the benefits of automation and software to all practices in the same way as we identified here. The study also did not collect data spent in the time and the effort associated with shipment of cryopreserved specimens. This was a miss on our part that, quite frankly, we would love to do over again when we have the time to collect data on shipment, since it is a big part of the operations of a cryostorage facility. We concluded that embryologists exert a significant amount of time, task redundancy, fatiguing work when they are working in manual cryostorage processes. There is a tremendous disparity of processes and space capacity across the different IVF laboratories. Integration of automation and technology solutions may address some of these issues. And finally, automation and software improvements should lessen the burden on embryologists, decreasing their fatiguing work and allowing for a more precise management of cryostorage operations. So with that, I would like to say thank you very much. And if you would like some more information on our studies and publications, please scan the QR code.
Dr. Collins, thank you very much for this comprehensive lecture. Indeed, the maintenance of the cryostorage facilities has a critical role in IVF labs, especially in ART centers with large volume of cryopreservation services. So my question would be, what are the most significant barriers preventing IVF labs from adapting automation and software solutions for cryostorage? Because, for example, in countries like mine, I'm from Turkey, and for example, cost effectivity is very important for whatever the technology that we start to use in routine practice. So would you like to comment on this?
Sure, I'd be happy to. And thank you for the question. I appreciate it. Probably the largest barrier that I believe most people would agree is that the current practices, the manual operation of the repositories, including a lot of the data handling, which is very centric on either written data records or sort of standard databases, Excel, or you pick your spreadsheet. A lot of those are difficult to adapt into these newer technology improvements that are coming. The benefit is that as the new technologies come on board and as automation comes on board, the field's going to move in that direction where it will be less and less of a burden to backwards adapt all of these cryopreserved specimens, which also leads to another issue that we have is that there are a lot of cryopreserved specimens that are out there across the entire world. And they do need management, they need constant management, and these collections are growing on a daily basis. So we need to be turning to more and easier and better ways to manage this, as opposed to a lot of physical labor and a lot of written and rudimentary databases that are liable for failure.
Okay, thank you very much for this nice lecture again. And it's my great pleasure and honor to introduce the second speaker, Dr. Lucie Delaroche, who is a senior embryologist leading the Bio Group IVF lab at Ramsay Parly 2 private hospital in Le Chesney, Roquefort, France. Her research focuses on the impact of disposable devices used in IVF on embryo development and ART success rates. She's an active member of the French ART Society.
Thank you, Basha, for this introduction. Good evening, everyone. I would like to start by expressing my sincere gratitude to RBMO for inviting me to present today at this webinar. It's a pleasure to share with you our finding on the assessment of bisphenol accumulation from disposable devices used sequentially in IVF routine procedure. Bisphenol or BP are organic chemicals commonly used in the industrial plastic production, either as building polymers for polycarbonate or as additive in epoxy resins. BPA, one of the most studied bisphenol, is known to impact fertility because it acts as an endocrine disrupting chemicals exhibiting a weak estrogenic activity. It has epigenetic effects on male and female reproduction with transgenerational impacts on the reproductive health of offspring. Due to serious concern about its safety, the use of BPA has been banned in the production of many items, such as baby bottles or food packaging. As a result, manufacturers have started replacing BPA with structural alternatives like bisphenol S or bisphenol F, which are not yet regulated. However, early studies suggest that these substitutes also negatively affect reproductive functions, although they seem to act through different mechanisms than BPA. Currently, culture media and consumables used in assisted reproduction are tested for toxicity to gametes and embryos. However, the standard tests, like the MEA, the mouse embryo assay, do not specifically screen for the release of bisphenol. Previous research has shown that disposable devices that we will call DDs used in ART procedures can release BP when individually tested. However, during an IVF procedure, several devices are sequentially used, potentially creating a cumulative exposure to chemical contaminants which could not be detected when single-use consumables are tested individually. So our study aimed to examine the release of bisphenol from DDs used in ART procedure and to determine if their level increase when multiple DDs are used consecutively under standard conditions. We hypothesize that testing DDs in combination would reveal higher quantities of BP released. The first step in our study was to screen 3 commonly used media and an oil for BP. For each test, a sample of medium was collected directly from the bottle without pre-warming. For disposable devices, we evaluated 19 different references commonly used in routine IVF procedures. These devices were first tested individually in 31 separate tests, and then we evaluated them in 9 different combinations designed to replicate the key step of an ART procedure. BP extraction was performed using pure water under conditions that mimicked routine clinical practice in terms of both duration and temperature. The release of 10 bisphenol was quantified using the isotopic dilution method combined with online solid phase extraction, liquid chromatography, and tandem mass spectrometry as previously described. The detection threshold varied depending on the specific bisphenols analyzed. So here is an example of a test that mimicked the process of sperm collection in a sperm cup, followed by its selection through gradient density centrifugation, freezing and thawing in high security straws. In this test, a specific volume of water was sequentially transferred between the 15 consumables to replicate the workflow. So our results. First, media. BPS was detected in all MHMC replicates at a concentration of 33 nanograms per ml. The hyaluronidase in verticult flushing medium contain both BPA and BPS with significantly high level quantified as 34 and 72 nanograms per ml respectively in all replicates. For liquid paraffin, 1 replicate tested positive for BPS at 2 nanograms per ml, while the other 2 showed only trace amounts. On the other hand, no bisphenol were detected in the CSEMC and CSEM-NXC culture media. As the Bergen laboratory, which performed the BPSs, lacked a CO2 and N2 gas incubator required for pre-gasing of culture media, the CSEM media were unsuitable for investigating BP release from individual DDs and their combinations. So pure water was selected as extraction medium for evaluating BP release from DDs. BPS was consequently detected in all 3 replicates of oosafe 100 millimeter culture dishes with a concentration of 32 picograms after 3 minutes of extraction at 37 degrees. This was observed from both the bottom and the lid of the dish. However, when the extraction was performed at room temperature for 13 minutes, no BPS was detected at the bottom of the dishes. Both PBS and PBA were consequently found in all 3 replicates of spermicide-free condom with concentration of 95 and 83 picograms respectively. And for CBS high security straws, BPS was detected in all 3 replicates as 3 picograms after 5 minutes of extraction at room temperature when the cotton in the straws was wetted. However, BPA was not detected or only in 1 replicate at 4 picograms when the cotton in the straw was not wetted. No bisphenol S, A, or other bisphenol were detected in any of the other devices individually tested. When disposable devices were combined, both BPA and BPS were detected in all 4 replicates of Association B, which included 13 disposable devices. This setup mimicked the sperm collection in condom, followed by selection through gradient density centrifugation. Moreover, BPS was also consistently found in all 4 replicates of Association C, which replicated sperm collection, selection through gradient density centrifugation, freezing and thawing in high security sperm straws. In Association D, involving 17 devices and replicating oocyte pickup and collection, BPS was found in all 4 replicates. Demonstrated that 3 out of 19 disposable devices released detectable level of BPA and BPS when tested individually. Among the 9 combinations of DDs tested, 3 released significant amount of BPS and BPS. The presence of BP in these combinations appeared to be directly associated with the 3 specific DDs that had previously been identified as major source of BPs. The amounts of BP detected in this combination were consistent with those found in individual devices, suggesting that there is no clear evidence of BP accumulation during sequential use. Our finding indicates that most disposable devices used sequentially in IVF procedure do not significantly contribute to detectable BP accumulation. However, certain consumables, such as the spermicide-free condoms, sperm straws, and specific culture dishes, stand out as consistent individual sources of BPA and BPS contamination. And included these BP-releasing devices could potentially lead to contamination throughout the entire procedure. In this study, none of the tested consumables were made of polycarbonate or epoxy resin, included that 3 that tested positive for BPA and BPS. Instead, they were composed of polystyrene, polyurethane, and ionomer resin. This makes the exact source of released bisphenol in these cases unclear. However, we did identify that extraction parameters, particularly incubation temperature, are critical factors in detected bisphenol, showing how sensitive their release is to these conditions. That said, the study has some limitation. The very low amounts of bisphenol detected, especially when only 1 replicate was found positive, raise the possibility of external contamination near the quantification limit. Additionally, the extraction methods didn't fully replicate the dilution that occur in IVF techniques, and oil overlay wasn't included. While water was used because it's free of bisphenol, it didn't replicate the full physiological environment, such as osmolarity or the complex composition of bodily fluids exposed to plasticware. To conclude, these findings emphasize the need to reduce bisphenol contamination in health media and devices. This could mean improving material selection, refining processing techniques, and prioritizing research into safer alternatives like BP-free plastics or even non-plastic options such as glassware. While factors like cost or compatibility with existing procedures are important, exploring this alternative is crucial. Finally, understanding how plastics affects health and reproduction is essential to shaping stricter regulation and minimizing potential risks. I would like to extend my thanks to Anne Togola and Sébastien Bristow for conducting the bisphenol essay at the BRGM laboratory, and Lucie Bénard from my lab for our collaboration in this study. And I appreciate your attention. Thank you.
Thank you very much, Lucie, for this great presentation and also for submitting this very interesting paper to RBMO. You know, your results are both encouraging and worrying. So one thing that I may have missed, and I wonder if you could address, is when you say significant quantities and you're measuring these concentrations in nanograms per mil, what is a significant quantity? What does it mean? Do we know what that threshold is where we have to start worrying about these concentrations?
Yes, thank you, Mina, for your question. It's an important question, effectively. There is no, actually, there is no threshold for BPs. However, we know that a fertile woman has a BP concentration from 0.3 to 1.2 nanogram per ml in her body, in normal, in her serum, urine, and follicular fluid. So if we are above this normal or environmental normal concentration, it could be critical, yes.
Right, it's very interesting, but I'm assuming that you will continue these studies and perhaps get into more the effects of the concentration related effects on development in your future studies.
Yeah, we try to study the cumulative exposure of plasticware by the MEA, the mouse embryo assay, and under our specifically routine conditions in my lab. So it is very, the experiments are very like we do in a routine procedure, and we will see with MEA if there is an impact or not in blastulation rates.
Great. Thank you so much. We will continue the discussion during the Q&A. In the meantime, thank you. I would like to move on to our next speaker. It's my pleasure to introduce Dr. Sarah Martins da Silva, who is a clinical reader in reproductive medicine, leading a translational research program focused on male infertility, the investigation of normal and abnormal sperm function, and drug discovery. Sarah also serves as an honorary consultant gynecologist, clinical lead for NHS Tayside Fertility Services, and HFEA person responsible for the 9 Wells Assisted Conception Unit. Additionally, Sarah directs the Scottish Sperm Studies Research Clinic, supporting couples affected by low or total failed fertilization following IVF ICSI. Sarah will be talking to us today, presenting her paper on artificial oocyte activation, improving ICSI outcomes. Sarah, please.
Thank you very much. And may I just say again, good evening to you all from a cold and frosty Scotland. And thank you to the organisers for asking me to speak this evening on a paper that we recently published in RBM Online. Now, I'm supposed to put up a slide kind of declaring my conflict of interest. As Mina's already mentioned, I'm an NHS consultant working at Ninewells Hospital Assisted Conception Unit in Dundee. I'm also a clinical academic involved in research, trying to understand how sperm work, but I'm also fascinated with fertilisation events. We hold grant funding from the Bill and Melinda Gates Foundation and Innovate UK, and I've received honorary and speakers' fees from various organisations, but I don't perceive that there's any conflict of interest. One last thing to mention is I'm also involved in postgraduate teaching. So if any of you are interested in following further studies at the University of Dundee, there are a couple of QR codes here for our face-to-face and also distance learning courses. So on to the kind of matter of the evening. We are all probably familiar with the Vienna and Marabou consensus, key performance indicators for lab performance. We know that not every egg will fertilise following IVF and ICSI, but we'd expect the majority of the eggs to fertilise, expecting somewhere between 65 to 80% fertilisation rate. So total failed fertilisation is estimated to affect somewhere between 5 and 10% of IVF cycles and up to 5% of ICSI cycles. There's also a further kind of group of patients that have unexpected low fertilisation, which is variously defined as either less than 25% or less than 30% of oocytes inseminated. And these events clearly impact downstream on IVF success overall. Now, fertilisation abnormalities are uncommon, but they're clearly very devastating both to the patients as well as the lab and clinic staff involved in these cases. So if we have a case with failed fertilisation, we kind of work our way, as I'm sure many groups do, through a series of questions trying to ask why on earth has this happened? And we look specifically at lab issues. Has there been any issue with the media, the pH, the temperature, any sort of equipment malfunction within the lab itself? We then review the cycle and think about stimulation, number of eggs collected, any issues with egg maturity or poor egg quality that's obvious. And we then look at the sperm side of the equation and think, was there, very, very abnormal morphology, globozoospermia, for example, or clearly very poor samples that have been used in treatment, perhaps frozen and thawed samples that are immotile, for example. And when you kind of go through this list of potential scenarios and explanations, you may then end up with this end diagnosis of unexplained. We don't really know what's happened, and then this is the part that we have been very interested in, because my theory is that probably nestled within this apparently unexplained fertilization abnormalities sit a lot of sperm function problems that we just simply haven't diagnosed by diagnostic semen analysis. We know that there are a lot of different functions that sperm require to bring to fertilization. You'll be familiar with the concept that immediately ejaculated sperm are unable to fertilize an egg. They need to go through a series of different events, which we term capacitation. And there's multiple downstream events and steps that the sperm need to do in terms of receptor ligand interactions and ion channel changes, membrane fusion, proteolysis, as well as oocyte activation for fertilization to occur. And we and others have published many papers looking at a variety of sperm-related causes that may underlie IVF fertilization failure, although most of these are circumvented by ICSI. So the sperm brings its DNA to the egg and fertilizes it, but we also have increasing recognition that there are epigenetics, methylation, post-transcription histone modification, and contributions of mRNA, as well as proteins that are required for fertilization and embryo development, which is very eloquently described in this diagram here. But one of the key features is oocyte activation, thought to be modulated by a protein termed phospholipase C-zeta. And the arrival of phospholipase C-zeta born by the sperm into the egg at the time of fertilization creates a wave of calcium across the egg cell, which is described as a Mexican wave, but then also initiates calcium oscillations modulated through calcium release from the endoplasmic reticulum and so on. And overall, it's thought that oocyte activation deficiency is the commonest cause of low or no fertilization following ICSI. So as Mina mentioned, I've set up a sperm studies clinic, a FAB clinic, fertilization abnormality clinic. We set it up probably about 10 years ago, and the idea was really to motivated by couples experiencing failed fertilization, to offer them the opportunity to participate in sperm research studies. So we went through this process of looking at events in the lab, looking at egg stimulation, sperm issues, and so on, and offered the patients the opportunity to participate in various sort of a barrage of tests on the sperm, looking at CASA kinematics, looking at Kramer penetration assays, calcium signaling, electrophysiology, as well as phospholipase zeta protein expression, and some DNA studies alongside with that. And so the study that we published recently was looking at overall the performance of our lab and those affected by failed fertilization. So across an 8-year period of time, we had performed 3,095 treatment cycles with approximately a 50-50 split between IVF and ICSI. And in terms of the couples affected by total failed fertilization, there were 63 following IVF, so that's 3.9%, and 60 following ICSI, so 4%, and a further small proportion of patients affected by unexpected low fertilization. So when we then looked at the cases affected by total failed fertilization and going through this algorithm of trying to kind of find some plausible reasons for it, overall about 2/3 of IVF cycles and 1/3 of ICSI cycles, we concluded that there wasn't really any obvious explanation for what had happened to explain and justify or explain what had happened with the fertilization. So when you have fertilization problems following IVF, it's most common that people will move on to ICSI. And clearly within this cohort of 63 patients, 23 of them moved on to ICSI. And as you can see there, the overall fertilization rate was 74.4%, so very normal fertilization rate as expected for ICSI. But unfortunately, within that was one couple who experienced recurrent total failed fertilization, so an overall rate in this small group of 3.8%. Now, some couples went on to do repeat IVF, having changed the egg stimulation and kind of female side of the cycle. A large proportion, about 1/3, decided not to undertake any further treatment, and some couples reorientated treatment towards donor eggs and donor sperm. Lastly, there were 5 couples that went straight to IVF ICSI with oocyte activation, and I'll come back to describe that in a little bit later detail. Treatment for options following failed fertilization following ICSI are probably much more challenging. I think as a clinician, it's quite difficult to know what to say to these patients about what to try next. One option clearly is that you just repeat the same thing again. They go through an egg stimulation, egg collection, and you repeat ICSI with the potential risk of failed fertilization all over again. Another option is that you could offer them treatment with using donor sperm, although we know that this is obviously a very different concept and actually not acceptable to all couples. And the third opportunity then is potentially to consider application of oocyte activation. First published case reports were in 2010. The commonest approach across the clinics uses a calcium ionophore treatment. And I would stress, I don't think that this should be used routinely by any stretch of the imagination, but many clinics have now adopted this for use in quite specific circumstances. So this is the media that we use. It's commercially produced by a company called Gynemed, and it involves really very simple. You kind of need to gas the media before you start. After you've done your ICSI egg injection, you then put the eggs into the media for 15 minutes and then through a series of washes. It's a fairly straightforward product to use. And then if you're using EmbryoScope as we would do, then you would then set up your EmbryoScope slides and put your embryos into or eggs into culture. But there are some concerns, not least because there may be cytotoxic effects relating to massive elevations of calcium within the injected eggs, and potentially kind of epigenetic alterations with downstream issues for embryos and for children born as a result of this technique. But so far, data published has suggested that there is no increase in chromosome segregation errors or any issues with meiotic divisions. And a small case series has followed children up to the age of 10 years with no developmental or neurodevelopmental issues identified, and no increase in birth defects in a larger study published in 2020. So overall, although the data is limited, there are no real red flags here. Our case series looked at, as I say, 8 years worth of treatment and selected couples that subsequently underwent oocyte activation with ICSI, following and comparing the outcomes of that to the previous cycle of treatment, either IVF or ICSI, that had been affected by total failed fertilization. The number of couples in the study is 39. The cycles were immediately before and after each other, if that makes sense. So there is actually no significant difference in female age, although you may understand that the women were slightly older in their subsequent oocyte activation cycle. In both cycles, there were similar numbers of eggs collected. There was no statistical difference in the number of eggs inseminated or injected. On the left of my screen here then is the fertilization rate for ICSI and for IVF for the patients that were included in this study. So IVF fertilization rate overall was 4.7%. That little green dotted line represents 25%, which was our sort of low fertilization cutoff rate. For ICSI, the overall fertilization rate was 8.1%, so a cumulative overall fertilization rate for this group of patients of 7.1%. The graph in the middle then shows the fertilization rate for the same patients following ICSI and then oocyte activation. And we had an overall average fertilization rate of 57.2%, so a significant increase in fertilization. And the graph on the right really shows a selection of dots. So each of the dots represents an individual couple. And as you can see, most of the dots are joined and go in an upward trajectory, but there are a couple where they did less well with oocyte activation compared to their preceding cycle. As you might expect, if you've got more eggs fertilized, then we saw a much greater number of couples that had a fresh embryo transfer, that got pregnant, and that had babies. And a lot of couples also had cycles with frozen embryos, and therefore the opportunity to conceive following frozen embryo transfer, even if their fresh cycle had been unsuccessful. And in total, we report 15 live births out of 39 patients, and no fetal abnormalities in that group, although notably one termination of pregnancy for a significant cardiac abnormality. For a subgroup of patients, we also studied the sperm in collaboration with the research group down in Oxford, headed up by Kevin Coward, looking at phospholipase CZ immunofluorescence. And where we identified significantly lower amounts or overall quantity of phospholipase C-zeta, these patients were directed or discussed about going straight to ICSI oocyte activation without having ICSI in between time. So for the 10 patients where we identified what we thought was a significant abnormality with phospholipase C-zeta, we saw a much greater uplift in their overall fertilization rate, so 66.3% fertilization rate on average for this group of patients. In summary, total failed fertilization affects up to 10% of IVF cycles, up to 5% of ICSI cycles. It has got a complex etiology. If you look at lab and technical issues, however, we had none in our study, and these are, I would think, in this day and age, very uncommon. Poor stimulation response, low number of eggs retrieved, poor egg quality is in theory modifiable by alteration of stimulation protocols or use of donor eggs. And I would argue in this particular study that actually sperm dysfunction is common and a reasonably common occurrence in this particular group of patients affected by fertilization abnormalities. So again, semen analysis doesn't look at sperm function in this way. We don't have a test for phospholipase C-zeta, but we have an unmet clinical need for a validated assay to screen for this. There are some concerns relating to safety and efficacy of oocyte activation, But I would conclude for this specific group of couples affected by apparently unexplained fertilization abnormalities that ICSI oocyte activation is a very powerful tool. So thank you very much.
Yeah, thank you very much, Sarah, for a great talk. And you may sit in cold Scotland, but you warmed my heart, because a gynecologist being interested in artificial oocyte activation is quite unusual. I really appreciate that. So one question straight coming to my mind, and that is, why did you apply artificial oocyte activation combined with ICSI after one IVF failure, which is quite a step forward, quite an invasive technique to treat?
Yeah, only for those couples where we'd done the immunocytochemistry and we had a plausible explanation that there may also be a deficiency in phospholipase Z2. We felt that that was enough within the context of the research study. We felt that was enough evidence to justify the additional intervention of oocyte activation. So it was 5 cases of IVF failed fertilization where we'd done the immunocytochemistry that went straight into oocyte activation.
Yeah, but you didn't have the chance to split the oocytes to do a sibling oocyte study.
No.
Okay. Yeah, but let's discuss later on. We move on with the program, and actually we finalize it. So may I introduce Mitch Schiewe, who is a PhD from US. He attained his master's degree in animal science, reproductive physiology at Louisiana State University in Baton Rouge, Louisiana, and his PhD in physiology at the University of the Health Sciences in Bethesda, Maryland, followed by a postdoctoral NSF associateship at the National Institute of Health, National Center for Research Resources. He has applied art research in animal models, conservation biology, and human IVF for over 4 decades, specializing in cryobiology. So please go ahead.
Thank you, Thomas. I need to back up on a slide here, excuse me. Thank you very much for the I3 Network and RBMO for inviting me to participate in this webinar today. And I want to extend a debt of gratitude to RBMO for a fantastic editorial staff that helped us with these papers coming to press. In our study, over 2 years ago, we designed a 5-phase study to look at the of introducing ultra-fast vitrification and a 1-step post-warming dilution into clinical practice for oocyte cryopreservation. In these 2 papers, we represent the first 3 phases of the validation and verification process that ultra-fast vitrification is indeed effective. In phase 1 and phase 2, we used a germinal vesicle model to assess both survival and post-warming viability as determined by oocyte maturation up to 48 hours. In the second phase of that study, we then looked at the viability of those matured oocytes in terms of the meiotic spindle as well as activated embryo development. And in the activated embryo development phase of that study, we did compare the straight ionophore treatment with the additional exposure to dimethylaminopurine for 3 hours to assess what might be the optimal treatment when we move forward with mature eggs. Now, in phase 3 with the mature eggs, we again repeated these comparisons that we are looking at with conventional vitrification to ultra-fast vitrification in the preliminary studies, as well as the dilution treatments being conventional dilution to ultra-fast or to rapid elution. And in all cases, in all experiments, we had sibling fresh controls in each group from the germinal vesicles. And in phase 3, we moved on to a substandard mature egg model where oocytes that are rejected for egg banking were deemed discard exempt materials for research. And in those studies, we continued our assessment of survival, meiotic spindle organization, and activated development to the blastocyst stage. The authors of these papers have no conflicts of interest to disclose. I'd like to acknowledge my collaborators, Catherine Wozniak and colleagues at the Asian Egg Bank and Hanabusa IVF in San Diego, California. Catherine is now back in her homeland in Australia, working in conjunction with Monash IVF. Also, a young scientist in my lab, Ryan Reichelderfer, was heading a project with other embryologists and the physicians in our group at California Fertility Partners and Santa Monica Fertility Laboratories in West LA, all part of the Pinnacle Fertility Network. As we all know, vitrification greatly improved oocyte cryopreservation, and by 2010, there were numerous publications promoting successful outcomes and healthy live births. In the fall of 2012, we're familiar with ASRM Practice Committee being inclined to deem oocyte cryopreservation as non-experimental, aimed to help women with medical needs. And in fact, in that practice document, they cautioned against elective freeze preservation because it simply was not as efficient as embryo cryopreservation at that time. And in fact, 10 years, 15 years later, it really still is not. And although progress has been made with donor egg banking, primarily based on standardized warming practices and the selective bias against poor stimulating or producing donors. Batch variations continue to be a prevalent concern and problem among autologous patients. Now, we all know that cytoplasmic preparedness of the egg is important and probably is a key source of batch variation in egg freeze cycles. The oocyte competence of embryo development is certainly influenced by both nuclear and cytoplasmic events. I've long believed that the cytoplasmic preparedness of the oocyte to cryoprotectant exposure, especially concentrated vitrification solutions per unit time, is indeed altered by gene activation events, organelle function, and membrane permeability characteristics, as recently confirmed in some really eloquent mouse model studies by Zhang and Cho. Now, in grasping the concept of ultra-fast vitrification, I think it's important to go back 40 years and just look at a notation by Bill Rawl in 1980 when he first looked at cryomicroscopy and the effects of slow freezing. He made the observation that if the cell, in this case embryos, were sufficiently dehydrated to near their freezing point, a point which the cryoprotective additives could bind with the colligative properties of the remaining water and proteins to form a viscous interior that would vitrify upon rapid cooling. Then, as vitrification technology came about, as he, a few years later, got together with Greg Fay, who is producing vitrification solutions. We know that those form metastable solids outside the cell, but still we have this highly concentrated cryoprotectant that dehydrates the egg. Now, the technology we have used for the last 20 years then uses an equilibration approach in which cryoprotectant continues to load into the cell. It swells full of this non-vitrifying cryoprotectant, and then a secondary dehydration step at the end with vitrification solution is imposed. The concerns here are the exposure to the cryoprotectants. Does it have an effect on the membranes and the genes and the organelle function? It was proposed about a decade ago in some mathematical modeling by Benson and Davidson that cytotoxicity related to cryoprotective additives could be potentially reduced by limiting exposure intervals in 2 or more steps. Miguel Gallardo, in his dissertation studies, undertook examining this cryotheory and putting it into in vitro application. And what Miguel found was that there was an agreement between permeability calculations and a responsiveness of the cell, and that they ultimately determined that the minimal exposure interval of 2 minutes was sufficient in conventional vitrification solutions to vitrify these cells. And in their pilot studies, they had 100% survival in their oocytes, zygotes, and embryos. These observations were subsequently confirmed recently in a mouse model by Cho et al. In Miguel's studies, Gallardo's studies, he had these eloquent shrink-swell curves. And what it shows in the upper panel is in the equilibration protocol, we see the initial dehydration of the egg in the ES, followed by extended exposure causes swelling. And then in the vitrification solution, we see again some dehydration occurring. And this approach has been referred to by Cho as a contraction, expansion, contraction model, whereas ultrafast vitrification, or what Miguel referred to as dehydration protocol, you see within 30 seconds in the ES that the egg reduces down about 45% in volume, with some equilibration of cryoprotectant into the cell to 60 seconds, at which point the vitrification solution causes a secondary dehydration with some additional equilibration. Since vitrification also includes the post-warming, we have standardized warming practices, but we have post-warming removal of the cryoprotectant, and there's been an increased interest in the last couple of years of one-step dilutions in the case of embryos. And this could be referred to as rapid elution because we're using a non-permeating sugar to extract cryoprotectant from the cell. Now, in the spirit of history, it's worth noting that this is not a new phenomenon, and that Stanley Lebo presented us the one-step straw in the early 1980s, where sucrose was put into the straw. Once it was warmed, it was shaken to mix with embryos, and then you could put it back into the cow directly or into a dish and pipette the embryos. The same approach was used by us and Dr. Rahl's lab in early vitrification work, in which we had successful survival and live births in sheep and mice. Now, in lieu of our studies, again, we had a 2x2 factorial treatments, as well as our fresh controls. And in the germinal vesicle model, we found a significant decrease in survival in our conventional vitrification groups, both in both dilution treatments. And the ultra-fast vitrification group was not different than the control, which did experience spontaneous degeneration in culture. When looking at the maturation, we found there was no difference at 48 hours in the number of mature eggs produced in each group, but we did see some significant delays in maturation in the conventional vitrification group, with more M1s and fewer M2s at 24 hours. In terms of the meiotic spindle, we saw no differences between the treatment groups, although if you looked at the retardant of the signal, the polarization or density of the light, we found that the ultra-fast vitrification group with rapid elution was more similar to the control than the other groups, which were significantly lower than the control. And in phase 2, experiment 2, where we activated these matured oocytes, we did see no difference in the ability to produce low levels of blastocyst production, but we did see, in fact, improvement in the activation treatment when we used the dimethylaminopurin inhibition of the cycle for 3 hours. Those studies were then continued in phase 3 to determine if that, in fact, was the optimal treatment to go forward with. In phase 3, we found again that the overall survival of the mature egg was higher in the ultra-fast vitrification group, having 100% survival compared to the conventional treatment, both at 3 hours and 24 hours post-warming. We found no difference in their activated development or in the ability to produce low numbers of blastocysts. Also in the mature egg model, we showed no difference in their meiotic spindle formation characteristics, but we did note that there was 100% reorganization of the spindle in the ultra-fast vitrification group. And finally, in comparison of these treatments in our substandard egg model, we saw 100% survival in the ultra-fast vitrification group, which was significantly higher than the conventional group at 92.5%, but no difference in their ability to artificially activate into embryos, with good levels of blastocyst production in all groups at the end. And the lower panel of the time-lapse imaging shows the normality and sequence of timing in embryo development over time in both ultrafast group and the conventional vitrification group. Overall, we found that ultrafast vitrification technology is a more efficient approach in that for the first time it offers the same reliability as embryo vitrification. Each step of our validation verification process has supported next steps, and knowing that healthy live births do occur following conventional vitrification methods when transferring blastocyst stage embryos, we anticipate similar outcomes in our ongoing comparative sibling oocyte clinical trials. And on that note, recent events reported out of ESHRE by Aiden et al. Have shown, in fact, that repeatedly high survival rates, excellent blastocyst production, and efficient clinical pregnancy outcomes can occur using this technology. Thank you.
Thank you very much, Mitch, for squeezing in all that information that we asked you to present in this short period of time. It was very informative. I just wanted to ask you a very general question before we move on to the Q&A and a deeper discussion, and that is, what challenges do you foresee in clinical implementation of your methods?
Well, I think one thing is, and I didn't have time to really cover, we did look at a secondary solution. It was a little bit different in type, and it had a little different responsiveness. I think every solution may have to be validated itself in terms of how we do ultrafast vitrification. We may find that 1.5 minutes or 2 minutes is more optimal in the ES solution than 1 minute. So there are some nuances we'll have to work out. We've certainly seen that in a blastocyst model. And then I think some of it has to do with, as it is more time efficient, it's, vitrification is a very labor-intensive technique. So we have to see, like in our case where we're doing donor egg banking, as in our experimental trial, can we do 2 batches of eggs at one time and do one at 1 minute, one at 1.5 minutes? So there certainly are some things to work out, but I think in the end it's going to be a very welcomed addition to the laboratory as far as workload and time efficiency. And I just think having the ability for all eggs to survive is something we've all been waiting for.
Yeah. I also hope we can talk a little bit about the GV egg as a model for this work, which is in itself interesting and important. But I'd like to invite our speakers and my co-host to join us again so we can start the QA session. And I will give an opportunity to Basak and Thomas to also ask questions. We have a number of questions that have been posed by the audience, and we will try to get to the hottest ones, I guess, in the next few minutes. Basak, would you like to start?
Yes, sure. Well, there are many questions that are addressed to all the speakers, so it will be a little bit difficult to pick the most interesting ones. Maybe I'll start with a question that was addressed to Dr. Michael Collins. Are there any particular aspects of your study that you feel require further investigation or validation?
Sorry, I had to check off my audio there. Yeah, thank you for the question. That is definitely the case. To begin with, we would hope. Sorry, I lost my video there for some reason.
We can still hear you, Mike.
Yeah,
there.
okay. Maybe back. Apologize for that, everybody. Yes, there are quite a few things. Number one, as I mentioned during the presentation, a big miss that we had, and we freely acknowledge this, is that we did not look at anything to do with shipment of cryopreserved specimens. I think that that's something that really needs to be investigated in the same manner that we investigated the other aspects of cryostorage management. In addition, I would like to, when we do some follow-up studies, and if other people take it upon themselves to do follow-up studies, I really would like to look at the work efficacy of the individual and how that impacts the time and motion required to complete a task, with the assumption or a hypothesis that the more efficacious individual, meaning a more tenured individual who's more confident in themselves, are likely going to be able to complete the task faster and also going to do it with less motion, do it a little bit smarter. I would love to see that correlation with those two. So definitely putting those two together, I think would be great. And then finally, we've done some other work looking at self-reported fatigue and somatization and burnout with embryologists. I would like to marry the two together to see if we can see if increased physical workload leads to more self-reported fatigue, burnout, and somatization with the embryologist. So really all of those being put together would, I think, in my opinion, help define the problems that embryologists are facing, and also, more importantly, help alleviate a lot of these work conditions so that they can perform tasks more efficiently with less potential for harm to themselves.
Yeah, thank you. Thank you.
Thomas, would you like to ask a
Yeah.
question?
May I stick with the AOA topic again? Because I have seen one general comment or question that I think would be a good start for a discussion. It comes from Hossein El Zaini from Victoria, Australia, because he stated that, and he's right, that AOA is not a new intervention. It's like 14 years on market now. And despite the accumulating evidence supporting its effectiveness, at least, also its safety, at least in part, I would say, AOA remains classified as experimental technique. And he asked, HFEA also has it as an AMBER technique or recommended under certain indications. And he's asking, what else do we need to make it a state-of-the-art technique or not call it artificial any longer? So Sarah, what do you think? What do we need?
I mean, interestingly, actually, the HFEA have now taken it off its add-ons kind of template. They consider it a treatment or add-on or additional technique that can be used for specific cases. I think it's all around justifying what you're doing. As I have said all along, I do not believe that this should be routinely applied to every cycle of IVF or ICSI. And I think that's the important part, isn't it? Is like that we need to recognize that whilst, you can't advance science without experiments, you need to recognize that, you've got to be careful what you're doing and that you're doing it in the right way for the right patients. I think it would be a much easier sell, if that's the right way to put it, to make it into sort of more streamlined practice if there was a way that you could identify which patients had a problem with phospholipase Z-zeta that were likely to have an oocyte activation problem. I think overall we don't really understand sperm function. There's a huge black box there about other stuff that we should be doing to test the man, and we don't because we don't know what to look for. So I think there's a much bigger question that kind of snowballs out from oocyte activation. There's increasing recognition now that there may actually be egg oocyte activation deficient problems, like there's a protein called WEE, etc. So that, there's so many different parts to this story. But I think it's interesting because when I most recently went onto the HFEA website, they've now taken it off that add-ons list. It's now a recognized technique, but has to be justified for specific circumstances. If you can't test the patients, if you can't justify what you're doing to the patients for right or for wrong, then that is the issue, isn't it? Is it until we get a test, until we know what we're doing with it, and we can justify what we're doing correctly, then I think it will remain on that sort of experimental list.
Yeah, absolutely. The proper way to do a study would be a prospective comparison. And then one arm of the study would not get a good method, maybe people are convinced of, and the other one would get AOA. So this is nothing that is feasible nowadays. And as you said, we should not forget that still it's not a physiological calcium pattern that comes with the ionophore. It's one peak and that's it. So it's not physiological. So it shouldn't be used for all, that's for sure. It's up to us to identify the subgroup of patients that might benefit. Yeah, thank you.
Because these are rare events, they're difficult patients to find, and constructing a study, how on earth would you do that? That's we've kind of looked at patients in real time, and in one clinic, which is a sort of a medium-sized UK clinic, we've amassed pretty carefully phenotyped patients, but we've only amassed 39 couples over 8 years of treatment. So it's a you would need a big collaborative effort. And I think that's part of our call in the paper is, we're very happy to share our data. And if other people either in the UK or overseas want to contribute data so that we can get a bigger data set to see if that helps inform things, then that would be a useful step forward. But I think ideally what I guess I'm kind of working towards is some sort of diagnostic that we could apply easily as part of diagnostic semen analysis, for example, to quantify or analyze phospholipase EZ, assuming that that's the candidate, and then maybe others as well.
Could you please have a look on cost effectiveness if you come up with such a test?
Yeah, well, for sure. And absolutely. But, it would need to, if you could do something that was easy, I'm sort of thinking of, like a COVID test, something that was easy and seamless that could be integrated within diagnostic semen analysis, where you're already looking at cells in a lot of different ways. That would be a real deal breaker, right, in my opinion. But, how near we are to that, nowhere close. The type of analysis that we're doing at the moment is flow cytometry, which is, we're lucky because our clinic is right next to a group of research labs, one of which is mine, but not very many clinics have access to that kind of experimental machinery.
Yeah. So Mina, do you have an interesting question?
Yeah, and a good discussion. I just want to say that the moral of the story that I take from you and Sarah is that you shouldn't apply AOA indiscriminately. And this has been advocated, actually, in the literature. And I worry about it a lot when I see that people say, well, ICSI is more successful if you activate oocytes. And so I think based on your discussion, your data, Sarah, and the fact that over this long period of time in a large center, you only found 39, you said, patients who you thought were candidates, potential candidates for this procedure. I think that's very telling.
We have done a couple of other cycles where we have had couples with globozoospermia. I think the data around that is probably fairly robust in the sense that a lot of couples will experience fertilization failure if they've got that, but it's not a very common pathology, certainly in Scotland. Yes. We've done a couple of, there have been additional cycles that we've done for other reasons, but what we were interested in is where you don't see a reason for fertilization failure. The hypothesis was there's a problem with oocyte activation.
Thank you. So I'd like to pose a question to Lucie from Xavier Polevia, asking if you tried the, in quotes, well-known microfluidic semen prep device for BPA release. I will probably, share my guess that this is Zymote, but then it could be any other device that involves what he says is the membrane used in the device is made of polycarbonate, which makes me nervous, he says.
That's
Lucie.
an interesting question. Yeah.
Yeah, very interesting question. Thank you, Xavier, that I know in France. I don't use the microfluidic in my lab, and I didn't tested it for bisphenol, so I can't answer your question. I'm sorry, but it's a good question, and we have to do to test it, and I'm sure that it is well tested with by MEA. And perhaps HSMA. So there are yet some robust tests for these devices, but effectively the question of bisphenol can be asked. Yes.
There's also, Lucie, a question from Simone Pallini in Italy, from Italy. And I think she's asking about whether these disposables that you used are medical devices in class 2A and CE marked. I'm supposing that this is a very European regulatory system question, is it?
Yes, it's a new regulation, MDR, that is more strict with validation of disposable devices. But I don't know if the industrial perform this kind of systematic test of the release of bisphenol. I don't know if it is performed systematically.
There's also a very good question from Shakaria Musa from South Africa. Is the release of BPs in the devices exacerbated by an increase in temperature?
Yeah, we've seen that in our study that the 100 millimeter dishes released some BPs when they were placed at 37 degrees, but they didn't release BPs when they were at room temperature. So yes, we suspect an effect of the temperature incubation of on this release. If we have to take measure to decrease this release, is perhaps not to pre-warm long time the disposable devices before use, but just a few minutes or maximum 1 hour of pre-warming is enough. And eventually you have to wash them, to rinse them before using to discard bisphenol. It's important, yes.
Interesting. Basak, would you like to?
Well, I'll have another question also to Lucie. Does storage conditions and usage protocols of disposable devices influence bisphenol release during IVF procedures? This is good for the lab practitioners to know, you know.
Yes, like I said, you should not pre-warm your devices too long. You choose rationalize the number of devices you use per process or per procedure. You choose, you have to choose and to know which consumables to use with documented certification. You have to use certified DDs and to sensibilize teams from the BP release, I think. Perhaps you should also prioritize eco-friendly packaging to avoid the BP release, because BPs are often in a packaging bag. So just a few advices like that for the moment. But I think guidelines should be more expensive like me. Sorry for my English.
Thank you.
Thomas, anything?
Yeah, I have a couple of short questions to Mitch, if I'm allowed to ask them. One is anonymous, most probably from Giles, because he's asking whether the type of cryo devices might have an effect on the thermodynamics, meaning an open system versus a closed system, for example.
Absolutely. Well, in this study, we use cryotops. I didn't mention that in the methods, but we've stuck with open devices because that's what's commonly applied to eggs. But it certainly is something, any of those who know me, I did a lot of work with a closed system called MicroSecure, which I believe greatly in. And I think that's next steps. I think we need to look at, these are the things we need to start validating next. I mean, we have years of research here, just as we did with the original vitrification approaches, that we can now start looking at the same questions we were asking before, but now with a new technology. So I do think it's going to potentially alter the times needed to optimize the outcomes.
Perfect. And again, one from Xavier asking whether you use a 1 molar or 0.5 molar sugar concentration for rapid elution, and if you use rinsing steps in the warming solution as well.
Yeah, no, in this case, in our practical application, we're just using a 1 molar sucrose solution, or it could be the TS, whatever the TS solution is in your commercial product, 1 minute, and then straight into the isotonic warming solution. We are not going straight into culture media. We are doing a ambient temperature wash first, and then we bring out, we do 1 minute and a wash drop, and then we stick it in a second wash drop, go get our culture dish, come back. So it's 1 to 2 minutes in that isotonic wash before it goes into culture media. I did see that question. And also the DMSO that we're using is just standard kits, which are 15% DMSO, 15% ethylene glycol. We would like to move away from DMSO, so we are going to be looking at some alternative non-DMSO solutions. But I suppose the negative effect of DMSO is even less of a problem now if we're going to be down to a 1 minute or 2 minute exposure time.
And there is.
And would the problem be pathogenetic activation, or what is the particular problem with DMSO?
My concern with DMSO mainly has to do with membrane dynamics. I think DMSO has been responsible for a lot of the membrane sealing events that occur in batch variations where it permeates in, but then it can't effectively permeate out because of the longer exposure. And so you end up with additional swelling and rupturing of the cells. But again, with ultra fast, we have so little cryoprotectant in there now. It really is probably not a problem because it's very easy to pull it out.
Thank you.
Well, there is a question from Hossam El-Zaini from Victoria, Australia, to the same speaker. Can we apply the ultra fast vitrification to freeze small number of sperm retrieved from non-obstructive patients?
I haven't tried it, but the answer would be yes, because basically we know that kinetic vitrification works with sperm because they have so little water in them. I suppose Dr. Isenchenko's the mother, both of them would be better to answer that question. He probably played with that, so.
Thank you.
There's a lot of work to be done with testicular biopsies and severe sperm samples like that, and this technology may help.
Mm-hmm.
Michael, there's a question from London, Julia Steiria. I hope I didn't totally mess up that pronunciation. She's asking, how much will the reorganization of cryostorage cost? Will it require relabeling of the samples? How does it comply with HFEA requirements on cryostorage audit? I don't know if you're able to address the HFEA requirements, but it is a concern, you know, what it takes to reorganize everything.
Yeah, thank you. And that's obviously an important and timely question. So to begin with, at least with what we're doing here, we do not relabel the specimens. We use the existing labels on them, so there is no requirement to do that. It would be very difficult, and I don't know if anybody's ever attempted to relabel something that's vitrified. It's next to impossible is what it comes down to. So we do not do that. Number two, about the HFEA, I'm happy to talk offline, but certainly everything we're doing is HFEA compliant, and we meet all of their regulations. I am not a regulatory guru, but I certainly know that we're able to do that. I think importantly, when it comes to cost, it's really a cost in terms of manpower and time and effort. And the majority of the cost depends almost exclusively on the quality of the current data set. And if that data set is very clean, if it has very few mistakes, if it's in really good shape, and you're able to one-to-one transfer the collection over, then it is very quick, it's much easier, and everything else. On the other hand, if the data set is in poorer shape, if it's a very, very old data set that has been sitting around for years, and we all know that data sets, regardless of what we think, there is drift that happens with the data over time, especially over decades. There's always a little bit of drift that's going to occur. Then that takes more time, and it actually costs more because it's more effort that goes into it. We have a process that we've worked out where currently it utilizes 2 of our employees with one of the center's employees to verify that the transfer of specimens are occurring precisely and exactly. The great benefit of this is that as you're doing this transfer process, there is the cost associated with it, but the great thing is that you're going through a full scrubbing of the entire collection. So there's that benefit that comes with this also. So.
Well, I agree with that 100%. You get to clean up.
Yes, exactly.
What's there and make sure that you have an accurate accounting of everything. Well, we are coming up to now 4:35 EST. And I want to ask Basak and Thomas if they have any burning questions. And if so, please go ahead and ask. And if not, we will move on to our thank yous and a closing of this nice session.
Okay, maybe I have a very last question to Sarah. How does artificial oocyte activation impact the epigenetic integrity of the embryos?
Well, I think the problem is we don't really know. As I've already alluded to, when you add calcium ionophore, you create a big surge, a sustained calcium increase that's like a table. Okay, so it goes up, it goes across, and when you wash it off, it comes down again. So that's, you know, you have that, and you've got the eggs in that calcium media with this high calcium level for 15 minutes. When you add a sperm or inject a sperm, normally what would happen is you have a surge of calcium that then falls and up and down, and though the timing of that varies between species, but in the human it's about 10 minutes for each peak and oscillation. So what does that do? I don't know. I don't think we really truly understand about how genetics and methylation and epigenetics and all this works. But clearly, if you, at that very early stage where fertilization events are just occurring, you change the environment and the calcium around it, it may have an impact. It's a theoretical concern, but I can't really answer it any more clearly than that, I'm afraid.
Thank you.
Okay. Thomas, did you want to ask anything else?
No burning questions from my side.
All right, then let me move on to the thank yous, many of which are due, and start really by thanking our audience for being here and for engaging and asking good questions. And then, of course, I would like to thank our wonderful speakers, Dr. Michael Collins, Dr. Lucie Delaroche, Dr. Sarah Martins da Silva, and of course, Dr. Mitch Schiewe. Their insightful and interesting presentations have made this session a great session, a great RBMO session. And I would like to also thank my co-hosts, Basak Balaban and Thomas Ebner, for their time, their enthusiasm, and really continued contributions to RBMO as section editors. And I do want to mention that I missed this at the beginning, that Sarah will be joining us as a section editor in the new year. Correct, Sarah?
Welcome.
Congratulations.
That's really wonderful news. Congratulations to you and to us for adding you to our roster of section editors. I would like to also thank Duncan Nicholas, who behind the scenes has organized this great session. And now I will pass it on to Thomas, who will continue the thank yous.
Yeah, thank you very much. Mina, the pleasure is on our side, I bet. Of course, finally, we should not forget the team of I3, the organizing committee of this initiative, and the people being in charge of that, who are Jacques Cohen, Peter Norge, Mary Ann Cervantes, Thomas Elliott, Frank Farley, Violetta Sura, and Charles Palmer. Thank you, guys. Thanks a lot for your work. Basak, up to you.
Well, I'd like to thank everyone who contributed for today's fantastic webinar. The last I3 session for 2024 will be on December 17th, which is specifically sponsored by Alpha Scientists, with highlights from the 2024 conference in May this year, which was in Lisbon, with special videos. Thank you.
And I'd like to invite everyone to register and attend that meeting and enjoy this beautiful film that we produced called Global Voices in Embryology. It truly is impressive, and I think you will enjoy it. So please join us next week for the last I3 session of 2024. And with that, we close the session. Thank you so much, everybody. Thank you, and bye-bye.
Thank you. Bye-bye.